Welcome to FunMET Database, your comprehensive guide to fungal secondary metabolites and biosynthetic gene clusters. This is the homepage view and guide through the database. First, we see an interactive maximum likelihood-based phylogenetic tree of all the available fungi producing secondary metabolites. You can expand it and click on a particular fungal species to have a quick view of their associated metabolites. After that, on this right-hand side panel, you are seeing a Sankey diagram for all the fungal species and their associated metabolites without going to another page. Below this, there is a word cloud of fungal species that represents chemical groups associated with them when you move your cursor on the names. Now we are seeing the quick search page of our database. To search, you can click once on the search box and a scrollable drop-down menu opens up. Here you can select your desired fungus, metabolites, or chemical group and click Search to view the associated metabolite, their 2D and 3D structure, basic taxonomy, and their possible target proteins and function. And finally, you can export the results. After the quick search function, we move to the download page of our database. With the same function drop-down menu, we have a default mode of search for only the fungus names. After choosing the fungus name, we can filter out the search with different options to filter from. After all these steps, we see the download result, which can be downloaded as a CSV, XLS, or JSON type file. Moving ahead, we have our Browse section equipped with 4 pages: Browse by fungus names, by metabolites, by habitats, and by BGCs. First, we see the Browse by fungus names page. On the left-hand side, we see a taxonomic classification of the kingdom fungi, which is further divided into Ascomycota and Basidiomycota groups containing all the available fungi we have. On the right-hand side, after choosing a fungus, we see fungal species and their morphological features. Associated chemicals and their 2D and 3D structures can be seen here. Moving to the next page called Browse by Metabolites, it comes with a simple layout of a list of metabolites available here and can be toggled between a list of 2D structures and 3D structures. To view the metabolites in detail, we recommend viewing the 3D part. Also, these browsing features can be filtered out with the panel on the left-hand side, which can filter out the search based on chemical group, fungus names, and molecular weight. The structure page gives more in-depth information about the metabolite, which can be further used in many other studies. After discussing the metabolite browse page, we move to the Browse by Habitats page. Up here, in the mountain scene, some trees, mushrooms, etc., you'll notice several interactive points, each one represents a different habitat. Simply click on any point to instantly view associated fungi and their metabolites in the results panel on the right. On the left, you'll find our handy search bar and habitat categories. You can type in a keyword or explore by checking categories like tropical zone or acidic forest. Now, we move to another major section of our database, Browse by BGCs. Here, users can search for a fungus by selecting its name from the drop-down menu. The biosynthetic gene clusters of these fungi have already been analyzed using AntiSMASH. Users can filter the results based on BGC types and specific clusters, allowing you to quickly pinpoint the clusters of most interest. As you apply these filters, the system dynamically updates to display only the most relevant data. After applying filters, you can view the HTML results on the next page, formatted similarly to the AntiSMASH output. This familiar layout helps you navigate and interpret the data with ease, regardless of your level of expertise. Finally, users can download the results, including genes, gene clusters, and the entire genome, with just one click. This comprehensive download option ensures you have all the data you need for offline analysis or integration with other bioinformatics tools. After this, we can check the About and Contact pages. FunMET DB aims to offer researchers an accessible and comprehensive resource for exploring fungal secondary metabolites and their biosynthetic gene clusters. Main features include a vast collection of fungal metabolites, detailed information on biosynthetic gene clusters, multiple browsing options for ease of access, regular updates with new compounds and data. FunMET DB sources information from trusted sources, including scientific literature, research data, collaborations, and genome sequencing projects. Whether you're a researcher, student, or enthusiast, FunMET DB is here to support your work in fungal metabolite discovery. Explore, analyze, and discover, right at your fingertips.